In lesson 2, we will elaborate on the principles of a resting membrane potential and then look at how neurons generate electrical impulses, both graded potentials and action potentials. We are now moving on to the physiology of neurons, how they generate electrical signals to communicate and perform their functions. So neurons are electrically excitable, and these electrical impulses involve the movement of ions across the membrane of the neurons through various channels and pumps. And the movement of charged particles changes the voltage across the membrane. So there are two types of electrical impulses. Action potentials are the ones that travel a long distance. And note that action potentials only occur along the length of axons. There are also graded potentials that are local and occur in the cell bodies and dendrites only. Action potentials involve the all or none principle, which means that once an action potential is generated, it will travel the length of the axon to the axon terminals. Also, action potentials have one specific voltage differential. So no matter how strong the stimulus, as long as it's strong enough to trigger an action potential, the amplitude changes in the voltage are the same for all action potentials. Whereas graded potentials can vary. Some actually bring the membrane away from generating an action potential. Those are called hyperpolarizing graded potentials, while others are more likely to lead to an action potential. Those are called depolarizing graded potentials. And again, they can vary in amplitude and only occur in the dendrites and cell bodies. So it's the flow of ions through ion channels and then the return to the proper side of the membrane via pump activity that create the electrical signals. In this overview of electrical signals, we see that neurons that are not undergoing electrical impulses are at rest, and the voltage difference across the membrane is called the resting membrane potential. Once a neuron receives stimuli, either via the dendrites or to the cell body directly, all those signals are integrated and create a graded potential. That graded potential may bring the membrane away from an action potential or bring it toward an action potential. If the stimuli are sufficient to bring the membrane to a magic value called threshold, that will then stimulate an action potential, and that signal will travel the length of the axon down to the axon terminals where it will trigger synaptic activity. The synaptic activity involves the release of neurotransmitters from synaptic end bulbs into the synaptic cleft, which will bind to receptors on the postsynaptic cell. That cell again could be another neuron or a target effector cell, either muscle or gland, and that's how the signal continues along the pathway. So neurons, like all cells, have different ion channels. Channels allow ions to travel by diffusion down their concentration gradient from one side of the membrane to the other. So either from the extracellular space into the intracellular space or from the intracellular space to the extracellular. But again, it depends on the resting levels of the ions that will determine the gradients for flow through these channels. There are different types of channels. Leakage are non-gated channels, which are always open and allow the ions to diffuse down their gradient without regulation. Now, nerve cells have more leaky potassium channels than sodium leakage channels, which means these cells tend to have potassium diffuse out of the cells more than sodium diffuses in. Recall that resting cells always have a higher concentration of potassium inside the cell. That's why potassium diffuses out, while they have a higher concentration of sodium outside. That's why sodium always diffuses in. So this results in the cell membrane having a greater permeability to potassium, and the cell loses more positive potassium charge than it gains positive sodium charge. This contributes to a negative resting membrane potential of minus 70 millivolts in nerve cells, meaning that on the inner surface of the membrane, the voltage is slightly negative, in part due to the fact that the cell is losing more potassium than it is gaining sodium. We also have ligand-gated channels. These open and close in response to a chemical stimulus, such as a neurotransmitter from another cell or some chemical. These often are the ones that trigger neuron excitability by opening, again, channels that cause diffusion of ions into the cell. If it's excitable, it's usually a sodium channel that opens to bring positive charge into the cell. Finally, we have voltage-gated channels. These open and close in response to changes in the membrane potential or specific voltages. So the flow of ions through ligand-gated channels then creates a change in the voltage, and at certain voltages, the voltage-gated channels open or close to then allow for or block the movement of ions across the membrane. Mechanically gated ion channels respond to mechanical vibration or pressure as their stimuli. In that way, they are similar to the ligand-gated channels in that these are the stimulus type of channels that will allow for the change in voltage that opens the voltage-gated channels. The voltage-gated channels are the ones that need to open and close during an action potential. Here we have depicted some of the ion channels located in the plasma membrane of neurons. Neurons have a lot of leaky potassium channels. When these channels are closed, they block the flow of potassium, but they randomly open to allow for the efflux or outward flow of potassium, because as you can see, the concentration of potassium is higher on the inner side of the membrane in the cytosol. To the right, we have mechanically gated channels. These respond to pressure and vibration stimuli. These stimuli open the channels, and in this case, they were allowing sodium and other ions to flow into the cell. Bottom left, we have ligand-gated channels. These channels have receptors embedded within them that bind specific molecules called ligands. That could be a neurotransmitter, it could be a blood chemical, etc. Depending on the type of stimulus, the binding of ligand may open a channel that allows potassium to flow out, or a different type of channel might allow sodium or calcium to flow in. So we're going to look at the effects of different channels opening or closing with respect to voltage changes in future slides. Finally, on the bottom right, we have a voltage-gated potassium channel. It is gated, meaning that it doesn't randomly open and close. It requires a particular stimulus to open it. These channels are closed at the resting membrane potential of minus 70. However, if the flow of ions through other channels change the voltage of the membrane, say to minus 50 millivolts, then that will open these voltage-gated channels. Potassium is then allowed to diffuse down its concentration gradient, in this case, out of the cell. So potassium always diffuses out, sodium always diffuses in, and the amount of each ion that flows in and out depends on the number of channels that are open, which is determined by the types and amount of stimuli. The resting membrane potential, or the RPM, refers to the voltage difference across the membrane when the cell, the neuron, is not being stimulated. The resting membrane potential exists because of a small buildup of negative ions in the cytosol along the inside of the membrane and an equal buildup of positive ions in the extracellular fluid along the outside surface of a membrane. This difference in charge creates a resting membrane potential of minus 70 millivolts, and because there is a difference, the cell is said to be polarized. Several factors contribute to the resting membrane potential. First of all, there's a difference between the concentration of various ions and substances inside and outside of the neural cell. The extracellular fluid is rich in sodium, we saw that before, as well as negatively charged chloride ions, while the intracellular cytosol is higher in concentration in potassium, as well as negatively charged organic phosphate and amino acids and proteins. So the negative phosphates and amino acids contribute to the negativity of the resting membrane potential. We also saw that the membrane permeability differs for sodium and potassium, where the increased number of leaky potassium channels increases the permeability for potassium 50 to 100 times. So again, the cell is experiencing a greater diffusion of potassium outward than the diffusion of sodium inward. So the cell again is losing more positive charge than it is gaining. And the inward flow of sodium cannot keep up with the outward flow of potassium. Finally, the sodium-potassium pumps in the membrane use ATP to pump 3 molecules of sodium out for every 2 molecules of potassium that are pumped in. So the pump is essentially removing sodium that diffuses in through its leaky channels as quickly as it comes in, while it is not keeping up with the restoration of the potassium that leaks out. So again, the cell has more positive charge being pumped out than positive charge moving in, and this contributes to the negativity of the resting membrane potential. Hopefully this diagram will help you visualize what's happening across the plasma membrane of neurons. So this shows you a resting membrane potential, a cell that is not signaling or experiencing a stimulus. The membrane has all of these proteins. So you have your leaky potassium channels, your leaky sodium channels, as well as your 3 sodium, 2 potassium pumps, and others that are not shown in this diagram. Let's focus on these. So the blue represents the extracellular fluid, the tan color represents the cytosol on the inside of the cell. On the left, we have a leaky potassium channel. Remember that neurons have a lot more of these than potassium channels, so the cell is constantly losing positive charge in the form of potassium as it diffuses out. The membrane has fewer sodium leakage channels, so it gains fewer positive charges in the sodium influx. Also, we have a high concentration of sodium outside the cell, as well as chloride ions. But inside, we have a higher concentration of potassium, as well as negatively charged phosphate and protein ions. Thirdly, the sodium-potassium pump on the right in purple shows you that these proteins use ATP to move 3 molecules of sodium out for every 2 potassium that are pumped in. This keeps the gradient for sodium high on the outside of the cell and keeps the concentration of potassium high on the inside. But again, the cell is losing more positive charge in the 3 sodiums that are pumped out than it is gaining positive charge with only 2 potassium being pumped in. So these are the conditions now that set up the negative resting membrane potential at minus 70 millivolts. Graded potentials are changes in the voltage, but they are small deviations from that resting value of minus 70. If they make the membrane more negative, they are said to be hyperpolarizing graded potentials. If they make the membrane more positive, they are said to be depolarizing graded potentials. Depolarization means that the membrane becomes less negative, more positive, that is usually due to the influx of positive ions. So these graded potentials are caused by various stimuli that now open channels in the membrane. They're called graded potentials because it can vary in a change in the voltage or the amplitude depending on the strength of the stimulus. They are also localized to the dendrites and cell body. Graded potentials can be created by the opening of mechanically gated channels or ligand-gated channels. Now, with either of these channels, they specifically allow ions to flow in one direction. And it depends on the location of the channel in the body. So for example, the binding of acetylcholine as a ligand neurotransmitter to receptors in certain neurons open up sodium ligand-gated channels where sodium, or in this case calcium, are allowed to flow into the cell. That is the acquisition of positive charge, so it makes the membrane less negative, more positive, and is said to be a depolarizing graded potential. In the bottom image, we have a different neurotransmitter, glycine, binding to a ligand-gated channel. In this case, it caused chloride ion to diffuse into the cell. Now, chloride ion is negatively charged, so when it diffuses in, it actually makes the membrane more negative on the inside and therefore has created a hyperpolarizing graded potential. Now we're getting to the main event, which is the generation of action potentials. An action potential, or an impulse, is a sequence of rapidly occurring events involving the opening and closing of specific channels that eventually depolarize the membrane and then repolarize it, thereby reversing the membrane potential and creating a blip, if you were, or a change in the voltage that is the electrical impulse. Action potentials involve the opening and closing of voltage-gated sodium and potassium channels in sequence, and these voltage-gated channels open at specific voltages when ions move across the membrane, initially due to graded potentials. So according to the all or none principle, if a stimulus is strong enough to depolarize the membrane to a specific value called the threshold value, that will open up the first set of voltage-gated channels, which will then lead to the opening of the next set of voltage-gated channels and create an action potential. If the stimulus is stronger than is what is necessary to reach threshold, it will open up the channels in the same way. The action potential is always the same, and once it is generated, it will propagate and cause opening and closing of the channels in sequence along the entire length of the axon. So a stronger stimulus would not cause a larger impulse. So here we have a graph that depicts what is occurring during the various phases of an action potential, which again occurs along the axon. On the left of the graph, we have a resting neuron at minus 70 millivolts. It experiences some stimulus, for example, the opening of a ligand-gated channel that allows sodium to flow into the cell. The influx of positive charge makes the membrane less negative and more positive and brings it up towards minus 55 millivolts. If the voltage of minus 55 millivolts is reached, the neuron has acquired the threshold value, and at threshold, voltage-gated sodium channels open first. This allows for a rapid influx of even more sodium, which further depolarizes the membrane up to a positive value of 30. So the voltage has flipped from minus 70 to positive 30. That's the reverse in polarity rather. All right, at positive 30, the sodium voltage-gated channels close, and voltage-gated potassium channels that open a little more slowly are finally open, and they now allow potassium to flow. But remember, potassium is going to diffuse out of the cell. So the cell loses positive charge and becomes more negative, and the voltage drops back down to minus 70. Often, these membranes experience hyperpolarization, where they overshoot the resting membrane potential value of minus 70, might dip down to all the way to minus 90, but then we have that after hyperpolarizing phase where the voltage-gated channels close and the sodium-potassium pumps that are always working move sodium back out of the cell into the extracellular space and move potassium into the cell, restoring the resting membrane potential values. So here we're going to compare the difference between graded potentials and action potentials. As cells experience smaller amounts of stimuli, if the change is insufficient to bring the membrane to threshold, it's called a sub-threshold stimulus. And when threshold isn't reached, the voltage goes back to minus 70. Again, those sodium potassium pumps are always working to restore and maintain resting membrane potential. So nothing happened. With a stronger stimulus, the voltage change will reach threshold. So it's called a threshold stimulus. And once threshold is reached, the voltage-gated channels open and close in sequence, leading to a change in voltage up to positive 30 and back down to minus 70. Now, if there's a really strong stimulus, it doesn't matter. It's still going to bring the membrane to threshold. The voltage will change in the same amplitude up to positive 30, back down to minus 70, even though there was a super threshold stimulus. So that's that all or none principle. Once threshold is reached, there will be an action potential, and action potentials always change by the same voltages. So before a neuron can experience an action potential, it has to be in its resting state. With the resting membrane potential, sodium channels are closed and sodium cannot diffuse in. This allows for the initiation of depolarization, where a chemical ligand or mechanical stimulus causes a graded potential that depolarizes the membrane to at least minus 55 millivolts, or the membrane reaches its threshold value. At threshold, voltage-gated sodium channels open quickly, allowing sodium to rush quickly into the cell. That influx of positive charge changes the voltage to about positive 30, and then these sodium channels close quite rapidly, within a few 10,000ths of a second. This now brings the membrane to the repolarizing phase. Note that when the membrane reached the threshold value of minus 55 millivolts, voltage-gated potassium channels also began to open, but potassium channel opening is much slower than that for the sodium channels. So during that time, depolarization occurred due to the influx of sodium through the open sodium channels. And when the potassium channels finally do open at around positive 30 millivolts, the sodium channels have now closed. So we no longer have the influx of positive charge making the membrane less negative, but the potassium channels are open, and the outflow of potassium now causes the membrane to become negative again, or it repolarizes the membrane down to the resting potential of minus 70 millivolts. Often, again, we said more potassium leaves the cell than is necessary, and the membrane hyperpolarizes to about minus 90 millivolts. But the potassium channels will close, the outflow of potassium will stop, and the sodium-potassium channels that are always working will return the ions to the proper side of the membrane and restore the resting membrane potential of minus 70 millivolts. Let's discuss this refractory period. You saw it a few slides ago when we had the graph of the events of an action potential. The refractory period is a period of time during which a neuron cannot generate or experience another action potential while it is currently experiencing an action potential. There are two types of refractory phases. The absolute refractory period is when the neuron is in depolarization and early repolarization, and even a very strong stimulus will not generate another action potential. Essentially, the cell has to reset before it can experience a new action potential. The sodium channels must return to their resting state before they can be reopened. Now, large diameter axons have absolute refractory periods of a whopping 0.4 milliseconds and therefore can experience a new action potential in a short period of time, leading to about 1,000 impulses per second. That is phenomenal. But that's the rate at which your nervous system communicates. Now, the relative refractory period is towards the end of the refractory period. This is when the sodium channels are closed, the potassium channels are still opening but are closing. The membrane is almost completely repolarized and maybe even hyperpolarized, and therefore a suprathreshold stimulus is able to bring that membrane up to threshold by depolarizing it. But it has to be a stronger stimulus. But again, once an action potential is generated, the amplitude changes will be the same. All right, let's look at how the action potential travels along the length of the axon. It is called conduction or propagation. Continuous conduction is how the signal travels along unmyelinated axon fibers. This involves a step-by-step depolarization of each and every region along the length of the membrane of the axon or the axolemma. So the opening of sodium channels here that causes depolarization leads to opening of the sodium channels in the next region, that leads to opening of sodium channels in the next region, and that's how it propagates slowly, continuously along the axon. It's like taking baby steps down the street. Whereas saltatory conduction occurs in myelinated axons, and sodium channels only open in the areas that are not covered by myelin, which are the nodes of Ranvier. And this is where depolarization and action potentials occur. So the current carried by the ion flow occurs from node to node instead of along the length of the axon. So again, this is like skipping down the axon as opposed to walking step by step down the axon. This is depicted nicely here. On the left, we have continuous conduction, where the current flow, due to the opening of sodium channels and depolarization, leads to depolarization in the next region, and then the next, and the next. Note that because of the refractory period, an area that is currently depolarized cannot depolarize again, so that the signal can only move forward or away from the cell body down the length of the axon. Now on the right side of the diagram, we have saltatory conduction, where the signal travels from node to node and skips across the myelinated regions. So it takes much less time for the signal to reach the axon terminals, and the signal travels or propagates much faster. There are several factors that affect the speed at which a signal will travel along or propagate or be conducted along an axon. We have looked at the amount of myelination, also axon diameter. Wider axons tend to propagate signals much faster than thinner axons. So the propagation speed of the impulse is not related to the stimulus strength, but to these other factors, where larger and myelinated fibers conduct impulses faster than unmyelinated, thinner axons. Remember that demyelination allows for saltatory conduction. Another factor that affects the speed of propagation is temperature. Temperature makes everything go faster. So increased temperature results in increased speed of propagation.